I'm Michael Reich, and I direct software development in the lab of Jill Messerov. And I'm going to tell you about our work incorporating Globus into our GenePattern Gateway. This software is developed in the lab of Jill Messerov, and we're at UCSD. And there are two arms to our lab. There's a research arm in which we develop statistical and machine learning algorithms for genomics analysis. And there's a software arm that's aimed towards supporting the analysis and visualization requirements of the worldwide genomics community. And in addition to GenePattern, the lab developed gene set enrichment analysis, known as GSEA, and IGV, which is the integrative genomics viewer. And these are all in the bioinformatics field. And in this field, these projects have been actively developed for over 15 years, and they have over 300,000 registered users. So regarding the GenePattern Gateway, this is a platform for reproducible bioinformatics research. It's a fairly long established project. The first release was in 2004, and at current there are over 84,000 registered users. We have a public server that runs in the Amazon cloud that runs between 1,000 and 5,000 analyses per week. And analyses can go anywhere from between a couple of seconds and a couple of days. GenePattern provides a repository of analyses that are somewhere in the ballpark of 250 analysis modules, and they include some of the most popular methods in genetics, genomics, transcriptomics, and proteomics, as well as general machine learning methods. For people who want to use GenePattern locally on their own resources, we also offer it as a Docker container. And I also want to note that this is an open platform, and many of the analyses that are available have been contributed by members of the community. So what does GenePattern basically do? GenePattern is aimed at scientists who don't want to deal with the installation requirements and resource requirements associated with running analysis tools on a local command line. GenePattern wraps these command line tools as web-based forms, and which removes the infrastructure details and allows researchers basically to focus on the analysis itself. The components of the GenePattern ecosystem include a repository of analyses and visualization tools, as I mentioned, an analysis engine that records all metadata about analyses that a user has run, including which version of the code was used for a given analysis, so that in the future, any result that is produced on GenePattern can be reproduced using the exact code, even if that code changes in the future. There's also a pipeline environment that allows scientists to chain analysis steps into workflows, and the workflows can be shared, reproduced, and also published. And one unique feature of this environment is that scientists are able to create a workflow in reverse. In other words, many times researchers don't know beforehand what the right steps of a workflow are going to be. And so we've implemented the ability to start with a result file and then create the workflow that produced that result file. All of these components are accessible through a RESTful Web Services API, and this allows us to present GenePattern in several interfaces. For programmers, we have a Python package, which essentially wraps the RESTful API, and that allows developers to access any GenePattern functionality directly from a script or application, and that provides the ability to use GenePattern as a back-end analysis engine to another portal, or just to perform the analysis within a given script. And we have also released a notebook interface in recognition of the success of the electronic notebook as the medium for reproducible data science. In 2017, we released GenePattern Notebook, and this is essentially a collection of extensions of Jupyter Notebook, and it allows scientists to add any available GenePattern analysis into a notebook. This gives you all the benefits of Jupyter, plus capabilities for bioinformatics analysis. And we also included additional functionality to make notebooks more accessible to non-programmers. So how does this look? Available analyses include everything that's available from the cloud-based GenePattern server, general machine learning approaches, as well as omics-specific analyses. Using GenePattern notebooks, you can add any GenePattern analysis as a cell within a notebook, and in this particular step, you'll see the ability to upload a data set, set input parameters, and run just as you would on the regular Gateway platform. A notebook can include analyses that are hosted on any number of servers, and this allows scientists to use compute-intensive methods on architectures that are best suited to their requirements. So Jupyter Notebooks has been mentioned in a number of the talks so far, and so as those of you who have to host them know, by default, the analyses run in the Jupyter kernel. And once you start having compute-intensive analyses running there, that can really interfere with the operation of the kernel. So we really want to be able to run compute-intensive analyses on resources that are best suited to them. The data transfer here is all done behind the scenes and is made seamless to the user. And the vision here is that a notebook can serve as a single document that provides a complete, executable, distributed workflow. And because our additions to notebooks are applicable in general beyond bioinformatics, I'll mention two additional features. One is the ability to add a single line of Python to a code cell, which will wrap it in a user-friendly interface. And this allows users to run the code behind that cell and change parameters as they would in a web form. So for end users of notebooks and users who are not that familiar programmatically, this allows them to use Python functions without having to mess around with the code. And it also makes notebooks look much neater. We also have a rich text editor that allows you to add text as you would in any word processor without the need to write any markup, HTML, or LaTeX. We host these notebooks on their own server, which is the GenePattern Notebook Workspace. This is a server that runs in the Amazon cloud, and it allows you to create, execute, and share GenePattern notebooks with the only requirement being a web browser. We also pre-install many scientific and statistical Python packages. And if there are any that users need beyond that, they can install them easily using pip. Users can share notebooks with other scientists and work collaboratively. And when notebooks are ready for publication, for example, as the in silico methods section of a publication, you can release them on the workspace and make them available to the public through a simple URL. Since we released this workspace, we've had over 15,000 notebooks created by over 4,200 users. And scientists who want to use GenePattern notebooks locally can also install it using the standard pip or conda package managers. So if you already have Jupyter Notebook on your machine, maybe through your Anaconda distribution, you can get all of the GenePattern Notebook functionality just by loading the GenePattern extension on top of your Jupyter installation. So given the number of interfaces and data endpoints I've just described, you can imagine that efficient data transfer of large files is a high priority. And this priority has been intensified by the growing size of genomic datasets. So compared with the talks that have preceded this, this is not going to be impressive, but the need to transfer data files of this size still presents a considerable challenge to people who are used to doing things through a web browser. So on the upper left-hand side, you can see that it was noted the relationship between the number of cells analyzed in an RNA single-cell-seq data set and the size of the resulting initial data set. And backing up here and just motivating the reason for this, this is one of the fastest growing genomics technologies, a single-cell RNA-seq data. The growth in this field has really been explosive due to new technologies. They allow us to measure the activity of genes at the resolution of a single cell. Whereas previously it was necessary to combine genomic material from a large number of cells in order to get a minimum volume of RNA for measurement, now we can query the genomic state of individual cells. And in the past couple of years, this has given us unprecedented levels of clarity in understanding cellular processes and how they relate to disease states. So getting back to the graphic, in around 2017, the high watermark was a data set of around 10,000 cells that corresponded to a little bit more than 100 gigabytes. On the lower left, more recently, you can see an atlas that consists of most major human cell types, and this was published in 2020. This consists of over 700,000 cells, and additional large-scale projects such as the Human Data Cell Atlas host single-cell data sets that are in the millions of cells. To support these data sets, we began a funded collaboration with the Globus team at the University of Chicago to incorporate Globus into the various GenePattern interfaces. The architecture of the resulting system is illustrated here. In the Amazon cloud, we host both our notebook workspace, which is a Jupyter Hub instance, and our GenePattern server on EC2 reserved instances. And we also have a GenePattern server running at the San Diego Supercompute Center that launches jobs on the Expanse supercompute cluster. We deployed a Globus endpoint local to all of these resources. And what we're seeing here is the original vision. So far, the actual deployment is a little different in that we found that a single GenePattern endpoint in the Amazon cloud servicing both the GenePattern server and the Jupyter Hub instance is so far sufficient. The San Diego Supercompute Center already had a Globus endpoint that we are now using for that server. And researchers who want to transfer data from local storage can use Globus Connect Personal. So how does this look to the user? We wanted to integrate data transfer with the UI to the maximum extent possible. And this was touched on in the previous talk as Rachana was mentioning, well, where you put features in your user interface has a huge impact on how much it's used. So the GenePattern user sees a Globus logo at the top of the Files tab. And when she wants to use Globus to transfer data, she will click on that. And she's then sent to the standard Globus web authentication tool to enter her Globus credentials. And when that's complete, the Files tab now shows the various Globus options, including the ability to transfer to or from a Globus endpoint. When the user selects either transfer option, the Globus file manager is launched. In these situations, the user only needs to select one Globus endpoint since the GenePattern endpoint is implied. We've also implemented a file download progress window that shows the status of in progress and complete file transfers, and also the ability to launch the Globus file manager from within the GenePattern interface. So what I've just showed you is the UI for the standard GenePattern server. We've also incorporated this into our Jupyter Lab-based interface. In the Jupyter Lab user interface, the Globus file manager is implemented as a side panel, and this is the common best practice for Jupyter Lab extensions. So Jupyter Lab users, if you told them that Globus was available within Jupyter Lab, that is exactly where they would look for it. Here we're illustrating the combined components of the Globus file transfer dialog within a Jupyter Lab panel. A and C here are the source and destination Globus endpoints, and B is a representation of the Globus file manager. And a user can click and drag a file directly from the file browser into an input box and have the data transfer done without the need to go to an external Globus file manager. We released the GenePattern server with Globus file transfer last fall, and that allowed us to continue to support the transfer of files that were already beyond the standard limits of browser-based HTTP and HTTPS transfers. And this will allow us to provide data transfer for arbitrary sized files in the bioinformatics field for the foreseeable future. The Jupyter Lab interface also is something that the Globus team is developing as a standalone Jupyter Lab extension. So those of you who host Jupyter Lab instances will also be able to take advantage of it when it's released. And these features were released within the past couple of months, so I look forward to being able to report on the huge volume of data that has been transferred via Globus in an upcoming Globus meeting. I'd like to thank you very much for the opportunity to talk and to acknowledge our lab members and our collaborators at U of Chicago, and I'll be glad to take any questions.